Hello, welcome back to the Philip J. Curry Dinosaur Museum Virtual Speaker Series, where we have paleontology guests call in for a presentation on science, fossils, and dinosaurs, and where the public will be able to ask questions both here at the museum and live on YouTube. For the viewers at home, you can leave your questions in the comment section, and I will read them to our speaker at the end of the talk. First off, we want to acknowledge that we are on the homeland of the many diverse First Nations and Métis people whose ancestors have walked this land since time immemorial. We are grateful to work, live, and learn on the traditional territory of Treaty 8. Today, Melina Jobbins will be presenting first African THYLACOCEPHALANS from a Late Devonian Lagerstätte. Melina grew up in the countryside of France, where from a very young age she started digging around her parents' garden for rocks and fossils. She completed her Bachelor's of Science in Geoscience at the University of Poitiers, France. She then moved to Bristol, UK for a year to get her master's in paleobiology under the supervision of John Cunningham and Phil Donahue. Thanks to a friend of the family, she got to meet Phil Curry and Eva Koppelhaus and did field work with their team for 2 summers in Dinosaur Provincial Park. She is now in Zurich, Switzerland, and is working on getting her PhD on placoderms from a new Lagerstätte in Morocco with her supervisors Christian Klug and Martin Rucklin. This Moroccan Lagerstätte is the same as the one she will be talking about today, containing a new mysterious alien-like creature, distantly related to spiders, lobsters, and crabs. Thank you, Melina, for joining us today. With that, I'll let you take it away.
Thank you so much, Cam, for inviting me. Hi everyone, thank you for joining to my little talk. It wouldn't be too long, so hopefully you guys can then get on with the rest of what you have to do today. So as Cam said, I started with my bachelor's in Poitiers in France because I come from France. I did my master's in Bristol then because I found that more interesting than the one they had in Poitiers being on mammals. That was something I didn't want to do. So I just moved on to do paleobiology so that it's more like general. There I did my master's dissertation on what we call the fossil embryos from Wang'an in China. These embryos are supposed to be the putative first animal embryos, although because it's not proven, we do lots of experiments on them. And my master thesis was on decomposing green algae to find out if maybe they could have a connection with the fossils. Then I moved on to Zurich, where I am now, and I'm in my second year, doing my PhD on placoderms from the Late Devonian in Morocco. You can see my 2 wonderful supervisors, Christian Klug and Martin Rucklin. Just so people can see quickly what the fossils I've been doing during my master's were about. Like these are the ones, the Wang'an fossil embryos. These were some of the algae I had fun decomposing. And this is a placoderm, which is what I'm working on now. So first I'm going to start with something a bit more basic for like the general public to know what I'm talking about, by starting with like explaining to you guys what is a Lagerstätte, which is a site which is really abundant in fossils. Like there's a ton of them. There's 2 different types of Lagerstätte. One of them that we call, because of the high abundance, concentrate Lagerstätte. So it includes, for example, bone beds or natural animal traps, such as the La Brea tar pits in California. The second type is a conservate Lagerstätte, which is about high quality. Usually, the ones that at least we know most about are, they do have a large quantity of fossils as well. But the main thing is that they have really good preservation. So it preserves like soft tissue, which normally we don't find, these kinds of things. 2 Canadian examples for the concentrate, so the high concentration one, bone beds. So of course, the one everyone knows, Dinosaur Provincial Park. Like for the ones who haven't been, you basically just walk anywhere in the park and you just have, like, you can't really avoid walking on fossils because there's so many of them. So you have like a few examples, like the triceratopsids, not ceratopsians, theropods, like lots of different dinosaurs that you can find there. And then the other type, the conserva, so the one like the well-preserved sites, including the Burgess shale, which Cam is doing an exhibit on at the museum. And now that you know what a Lagerstatt is, we can move on to Morocco, which with another well-preserved Lagerstatt, which is called the Thylacocephalan layer, which was described very recently by the previous PhD in the university I'm in now. In this layer, you can find like lots of Thylacocephalans, which I'll explain what this is afterwards. There's also vertebrates, mainly sharks. This is like an example. And placoderms, which is why I'm working on these now, because we do need to describe them at some point. This is the site, one of the sites, that's really abundant in these Thylacocephalans. The place is called Madinah El Mrakib in Morocco, and the layer, hopefully you can see the mouse, is the lowest like line that we can see that forms on the image. And this is basically a ton of nodules. Like you can see them on the pictures on the slides, or basically every nodule almost has a Thylacocephalan or remains of Thylacocephalans in them. The locality dates back to the Flaminian, which is Late Devonian. So you can see like somewhere around 370 million years old. And what are Thylacocephalans now? So the group is described with all these different features. The main ones being, so the bivalved carapace, which encloses almost entirely the body. So it goes like this. And just the bottom part, let's say, is open. It has at the front this typical rostrum on the carapace as well, which you can see then the eye goes underneath. Like it has very large compound eyes. Compound eyes meaning the eyes that you see in arthropods. And it has 3 pairs of appendages, so limbs in the front, which look a bit like crab-like, let's say, legs. And in the back, you have at least 8 pairs of extra limbs, which are much smaller and, as we describe them, paddle-like. They're like a bit more flatter and very much shorter than the other ones. Another typical feature is the set of 8 pairs of laminate gills, so gills for breathing, and laminate meaning that it's leaf-like. It's like very thin. They're arthropods, most likely crustaceans, but we don't have definitive proof, although it's been accepted by most researchers now that they are crustaceans. We can't tell what crustacean, like what specifically, in which group it belongs, because we need soft tissue for this. And the soft tissue we need is just not preserved for now. We haven't found anything. They lived from the Silurian, so you can see about like over 400 million years ago, until the Cretaceous, about 80 million years ago or so, in that case, and had lots of different shapes. Like here's just a few examples of the ones we know of, very few of them. The one at the very bottom with the black background, with the tiny eyes, this is actually the oldest one that we know of for now, the one from the Silurian. And then they quickly grew to having these huge eyes. They aren't known from many places compared to other groups, but when they are known, they're quite abundant, like in, for example, the Thylacocephalan layer. And this is Morocco. So this is like the first record of them on the African mainland, and the second one from, let's say, Africa. The Madagascar specimens being like over 200 million years younger. Ours is like really old. So this is what the holotype looks like. So the one we used to base the description on. You can see like the left image to help you a bit for the ones, because we don't always see really well, especially when we don't know it. We have the carapace with the outline on the carapace on the right, along with the eye, which is something we saw like at the start. It was just a nodule, which was given to me by my supervisor. I started preparing it, and then we just ended up finding, oh, there's a nice shell-like thing. And then we just kept on going, found the eye, and then CT scanned it, and then I have really cool stuff to show you just after. So I'm not going to go further. We named it, I got to name it like the species. The genus belongs to Concavicaris because of the typical posterior part of the carapace, which is very gently rounded. This is like a typical feature of the genus. And then we called it, I called it submarinus, which is Latin for submarine, because to me, with the reptoidal appendages, they do remind us of submarines, some of these weird looking submarines in movies. And its typical features are the ventral margin that curves outwards. You'll see it very well in the next slides. And this depression that you can see that I drew on the image, on the one on the left, B, it's like a very long depression on the carapace. You can see on the right image a close-up of the eye. For some people who want to see like the typical arthropod eye of in THYLACOCEPHALANS, with a very big close-up on B. And on C, this is actually the cuticle. So what you can see from very close on the surface, let's say, of the carapace. So we CT scanned it because we have the eye, and in that case, we hope to find more, because usually the eye isn't preserved because it's also part of, like I said, the soft tissue. So we CT scanned it, brought it to the computer, segmented it with a software, and we obtained this 3D model, which shows that the specimen we have has both its eyes, although slightly shifted simply because of taphonomical reasons. So like when the rock bested over it as it was fossilizing, everything just shifted a bit and it was slightly deformed, but that's absolutely normal in like lots of fossils. So it has both its eyes, which is really cool, but it also has most of its back legs that actually popped out, because normally they're much more inside, let's say, the shell, and only a bit of it comes out. This time it's like most of it's out. And we also have structures we noticed inside in the body. So we have on the top in red some very like thin structures that seems to like go around, let's say, something, but that thing is like not here. But when looking at other papers, we noticed that this is surrounding the place where the stomach is expected to be. So we interpreted this as the gastric muscles. We have then in blue these 3 pairs of like very thin sections, that's like very thin structures that are quite slightly elongate, but not as long as you'd expect them, let's say, in gills. But these are probably it, because they have the exact same structure as the ones we know of in other THYLACOCEPHALANS, but we just don't have 8 of them. We just have they're not entirely preserved, which is okay, that happens also. But we have 3 partial pairs preserved. And then we have this very enigmatic structure, which we haven't seen before in THYLACOCEPHALANS, like in the other ones, because they weren't preserved. And this is like in green, the one in green. This is a paired structure, very elongate, very close together. This took lots of time to figure out what this could be. Lots of research in papers, including arthropods, and then just going into crustaceans as well. And then we ended up with 2 main suggestions. The first one being the gonads, so ovaries in that case, being because some decapods, so a group of crustaceans, it is known that some of them have basically in that position in the cephalothorax, so between the head and the rest of the body, this pair of ovaries located very much in the back, just like here, and also very elongate, very similar shape, which is why this seems like a good interpretation for it. The other suggestion was the hepatopancreas. The hepatopancreas is something that is like, for us humans, it's the combination of the liver and the pancreas. This is what they have in arthropods. It's like both combined into the hepatopancreas. And it usually is located on top of the stomach, which is basically what we have here. The only thing is, we know from other Thylacocephalans that the hepatopancreas is actually really big compared to here, and is also located very much ventrally, so much lower, basically on nearly under the stomach in the case of Thylacocephalans. So this one is a big maybe, but we still keep the hypothesis because we never know. Even though, and the gonad is also something we don't expect to find ovaries in that position. It's rare, we have some examples, but it would still be a bit unexpected. But these are like the two main interpretations we have for now. So what can we tell about habitat and the diet, also with what we know from other Thylacocephalans? So we know that some of them had specific structures with the eyes and habits that let scientists think that they lived in deep, dark environments. Although this is controversial in some cases, like we aren't sure ours actually was, because it probably lived in the water column. And usually in the water column, it's not necessarily really, it's not like really dark. So some could have, some not. We know from some specimens, not the Concavicaris submarinus, but from other Thylacocephalans, that some of them hunted on small prey, for example, conodonts, because they have some leftover teeth in the gut content, or also hooks from cephalopods. This is also something else. If you don't know what a conodont is, you have a picture of it. And then with also, so we know that it was, at least in some places, a good food source for sharks, at least sharks. From the Cleveland shale, for example, there's a really good specimen that has some of the stomach preserved. And in there, you could find some of the shells of Thylacocephalans. So this could be an idea for our specimens as well, because we know that we do have sharks in the same layers. We have placoderms, and these are extremely abundant. So it would be like a good food source for them to feed on. They also have like these huge eyes. There was of course quite a bit of discussion about what was it used for. It most likely was used for predation, because they have a really large view, and they can see at the bottom, they can see from the sides, not from behind, but almost. And not only could it be used for predation then, but it can also be used to detect predators really well, because the only view basically that they don't have is mostly from above, slightly behind above. So that like it limits the possibilities for the predator to catch him. However, we know that given the back legs, because they are so small, this is usually what's used for swimming. Because they are so small and not very, like, it wouldn't allow them to have a very powerful swimming. So they're probably not very good at swimming. They just go around, chill. And for being a predator, this is not really easy to handle. So they have to adapt. And what they probably did was simply ambush the prey. Like they would hide somewhere and then just grab, like snatch any food that they can find. Like just something walks by, snap, there we go. So as a small like recap, we have like the first Thylacocephalans from the Late Devonian of Morocco, from which are the first ones from the mainland of Africa, and also the oldest African record. The holotype of Concavicaris submarinus, which is a new species, has preserved structures which were undescribed in Thylacocephalans before. So the gastric muscles, putative gastric muscles, and the green paired structure, which we interpreted, the one we interpreted as gonads/ovaries or hepatopancreas. And Thylacocephalans most likely lived in the water column, hunting for their prey through ambush, and were probably a good food source for these early gnathostomes, or early vertebrates like sharks, possibly placoderms. That's it for, as I promised, I didn't keep it too long. Like, just for future research, if people are interested in placoderms, I'll be like a new paper on this jaw piece of placoderm. So you can see with the arrow, it's actually like there's an anterior part and a posterior part in placoderms. And this is the posterior part of the upper jaw. And this paper is going to be published very soon, within like the next month, in Swiss Journal of Paleontology. We discuss inside like elements that we also preserved, including dentinous tissue, a pulp cavity, and such. But I won't say more, because if people want to know, they can also have a read. And then what's next? More placoderms for my PhD, more descriptions, lots more descriptions to do, because it's on because Lagerstatten was only discovered like 2 years ago, like described officially 2 years ago. There is still basically everything left to do with it, with all the descriptions of the specimens. And then if people want to come and have a chat during conferences and all, please do join, because we do tend to go to quite a few conferences, like PALAS, SVP at least, early vertebrate, etc. And if you have questions, please ask. If you have some popping up later on, feel free also to contact me through ResearchGate or the university website with my email. And also for like collaborations or any ideas you have, please feel free to just drop in an email or message. I'll always answer. And that's it. Thank you very much for your attention. I hope you enjoyed it. And if you have any questions, please do ask. Feel free.
Well, thank you very much, Melina.
Thank you.
Really appreciate this. All right, so we'll now move on to our question period. And if anyone has any questions, they can type them into the comment section. Our first one is from Jonah Weiss. Hi, Melina. Thanks for presenting. I have 2 questions. One, did your decomposition research project help build on this project, or were they entirely separate? Secondly, sorry if you already said, but what were the size of these creatures?
Oh, yeah, I didn't think. Hi, Joe. So for the first question, this is like 2 very different projects. Although for the first one, for my master thesis, it was taken over by his master student after me, Emily, and she is now publishing a paper with me as a co-author on in Sciences Advanced. And that's basically it for now with this, because now I moved on to placoderms. She keeps on working on all these things. So I'm hoping she gets to do a lot more afterwards as well with decaying. She can have fun decaying in the lab. I'm done for this for now. And the second question for the size of the Thylacocephalans. So it's, in the case of mine, the average was between like 5 and 6 centimeters long. Mm-hmm. The longest one we have in our specimens, the ones that we prepped anyway, so we know, was about 10 centimeters long. And the smallest one was like between 2 and 3, like 2.7, which is so cute. Wow. And for like Thylacocephalans in general, most of them are quite small. Like I think mine is actually like relatively good in size compared to lots of them. So most of them are like just a few centimeters, some less also, but generally a few centimeters long. So it's not very big. Not bad. We don't have a question, but a comment from Martine Watts. Thank you, Melina. That was very interesting. Got a smiley face. Thank you.
someone from the museum here was wondering, after placoderms, do you have thoughts on revisiting THYLACOCEPHALANS, or?
I would love to go back and do projects on Thealocaris, no matter when. I'm always up for it because learning about them was amazing. I really enjoyed writing that paper on them. It was really fun. I actually that was probably the most fun I had so far. Sorry, Christian and Martin for the placoderms, but I still love them. I love both. But it was really interesting to learn about a group that no one knows about, or very few people know about, and I would be really happy if I could get to do more with them at some point.
Kind of humbling. Do you have any thoughts about doing any more research, but less marine, more terrestrial vertebrates in the future?
Yeah, I don't have anything in mind particularly, but that's something I would be open to as well. I'm not particular, the only group that kind of doesn't really interest me, sorry everyone, is mammals. It's just a bit too recent for me, I guess, maybe, or too close to us, I don't know, but it's I would go to reptiles or something, for example. Reptiles I think are really, really cool, and that would be something I'd be interested in learning about as well.
Okay.
At some point.
We do have another comment from YouTube. Friedrich Lerouge, I hope I didn't butcher that. Thanks a lot for the talk. Very interesting critters. I have yet to encounter them in my Moroccan material. Are they found in other sites or just in Lagerstatten depositions?
So we still need to look around because it's quite recent. So because they are hidden in the nodules, most of the time you don't actually see them. So that's why that's why it's only been discovered actually really recently. My supervisor basically a few years ago just smashed a rock and oh, there's something in there. So that's how we ended up getting more, working on them. So we know that the layer is rich in them. It's outcrops in different places. The main one being Madinel Mrakib. There's other ones though, but I cannot list them just because it's really complicated Moroccan names.
Sure.
So I'm sorry, but if the person is interested, I can we can also just chat via email if you want. That's
absolutely and
fine.
that was Friedrich. We do have another question coming from Bruce Kosagi. With such large eyes, would these have been night hunters?
So there was a discussion about them maybe doing this, or at least hunting because of dark places. There was suggestions about it, but there's nothing certain because not that much. There's only one or two papers that actually looked well into the eyes to see if the structures were actually compatible with hunting in the dark. And so far it's, let's say, criticized. It's really not certain that they actually could do this, hunt in really dark places, so at night. It's more likely that they would I guess hiding behind stuff, just sneak attacks, these kinds of things, more than hunting at night. But I mean, why not? I don't know more than that because I didn't look also into the eyes myself. It's something I wouldn't mind doing at some point, but because we only have one specimen, we would have to, kind of ruin the eye to do the test, and that would be sad and heartbreaking.
Reconstructive sampling.
Exactly. It's ooh.
Friedrich also has another comment. And as a trilobite enthusiast, I just have to ask, are there any trilobites found at the site with this level of conservation?
So I don't think that there is, I haven't heard of that. So luckily I haven't gotten to see the site myself yet because I was supposed to go this year and COVID happened. Sadly. So hopefully I can say more within a year or two when we can go. But there is a paper from Linda Frey who talked about the whole alpha diversity, including in that locality. I don't recall there being any trilobites in there. So if there is, it would be extremely rare. The main ones are mostly THYLACOCEPHALANS. There's some mostly invertebrates, such as like I think it's like bivalves, gastropods, these kinds, more than trilobites. At least in this layer. Sorry.
Good answer though. Another comment from YouTube. Tim Hay, very nice talk, Melina. Smiley face.
Same.
I don't believe there's any more questions coming in at this time. So I think we will conclude our question period for now. Join us again for our next virtual speaker series on Saturday, January 23rd, 2021 at 3 p.m. Presenting from Southern Alberta at the University of Calgary, PhD candidate Jared Voris will be giving a special talk on all the different tyrannosaurs found in Alberta, titled A Dynasty Recorded in Stone, Alberta's Fossil Record of Tyrannosaur Dinosaurs. Thank you again to Melina and to all of you for tuning in. Have a safe holiday season and take care.
Thank you.